Hello students, this is Dr. Ranjini, Assistant Professor in Biotechnology. Welcome to e-learning sessions in immunology. Today in this session, we learn about the structure of antibodies. Antibodies may be defined as the proteins that recognize and neutralize any microbial toxin. They recognize and neutralize any microbial toxins or foreign substances such as bacteria and viruses. The only cells that make antibodies are B lymphocytes. Mainly, two forms of antibodies exist. One, those that are membrane bound, they are attached to the surface of the B lymphocytes. They are called as the membrane bound antibodies. The another group is the secretory antibodies. These are secreted to the body fluid. So they will be freely floating inside the system to recognize the foreign substance or the toxins which are there inside the host organism. The substance or the molecule that is identified by the antibodies that can evoke the antibody response is called an antigen. Is called an antigen. Some commonly used terminologies while explaining the structure and function of the antibodies are the serum. Serum. So, when the clot formation takes place in the blood, when the blood clots, the residual fluid that remains in the clot along with the clot, that fluid contains the antibodies. So these antibodies in the residue form the serum. So these antibodies will be there in that fluid along with the residue is called the serum. Next we use the term antiserum. Antiserum means serum contains a bunch of antibodies. When these antibodies show specificity to a particular antigen by binding to it, so those antibodies are known as antiserum. So in the pool of antibodies, the specific antibody binds to the particular or the specific antigen, that is called the antiserum. So the binding of the particular antigen to the particular antibody is called antiserum. Next is serology. Serology may be defined as the study of the blood serum or antibodies and their reaction with the particular antigens. So these terminologies will be used during the immunology. Next, coming to the structure of the antibodies. Antibodies are also called as immunoglobulins. They are called as immunoglobulins. They are represented as, there are 5 different types of antibodies. They will be represented as IgG or IgA, IgD, IgM and IgE. So in all this, so this Ig is common, immunoglobulin. That is the another name used to designate antibody. These antibodies are Y-shaped structure. They are Y-shaped structure, consists of two identical light chains. Consists of two identical light chains and two identical heavy chains. They contain two identical light chain and two identical light chain. Amino terminal region of the antibody. Usually, if for any protein, we will be having the amino terminal having the NH2 group and the carboxy terminal having the COOH group. So, the amino terminal end is called as NH2 and the carboxylic, that is COOH group containing end of the polypeptide is called as C terminal. So, these are glycoproteins. They are having the N terminal and the C terminal. Both your heavy chain will be having N terminal and the C terminal. Even your light chain will be having the N terminal and the C terminal. The amino terminal regions are called the variable region. So the around 110 amino acid residue region is called as the variable region. And the remaining region is considered as the constant region. The remaining region, the terminal or the C-terminal region of the immunoglobulin is called the constant region. And here the binding of the antigen to the antibody takes place in the variable region. They show the specificity towards the various antigen by showing the variability in the arrangement of the amino acid. Whereas the effector mechanism or transducing the signal to the rest of the cell or to the monocyte or to the other adjacent cell is mediated by this carboxy terminal. Is mediated by the carboxy terminal. Both heavy and light chains are composed of immunoglobulin domain. The Ig domain is a protein domain that consists of folded repeating units of 110 amino acid residue in length sandwiched between the two layers of beta pleated sheet. They are sandwiched between the two layers of beta pleated sheet. So here is, so there will be formation of the loop of around 110 amino acid residue and they are held together by the disulfide linkage. The two layers of the beta pleated sheet are held together by disulfide bridge or the disulfide linkage. There are short loops that connect the adjoining strands of each beta sheet. Amino acids in some these loop are most crucial for antigen recognition. The light and heavy chain structure is almost similar. Light and heavy chain structure is almost similar. In light chain, there is one variable region. In light chain, there is one variable region and one constant region. There is one variable region in light chain and one constant region in case of the light chain. Whereas in case of the heavy chain, the variable region is also one comprising of around 110 amino acid residue and the constant region may be varying from 3 to 4. 3 to 4. In this example, we find three constant region. One is constant CH1, that is constant region of heavy chain, and constant region of CH2, that is constant region of heavy chain, and another one is CH3. But in some antibodies, we will find four constant region. We will find four constant region. So, in case of heavy chain, the constant region may vary from 2 to 3. 3 to 4. Antigen binding site is formed by the variable region of one heavy chain and the adjacent variable region of one light chain. The disulfide bonds are formed between cysteine residues connect the light and heavy chain. So the light and heavy chains are held together by interchain disulfide linkages. They are held together by interchain disulfide linkages. These are formed between the cysteine residue. The association of heavy and light chains occurs partly due to the non-covalent interactions between variable region of light chain and variable region of heavy domains and between the constant region of light chain and constant region of heavy domains. The two heavy chains of each antibody entity are connected covalently by disulfide bonds. In IgG antibodies, disulfide bonds are formed between the cysteine residues in CH2 region. So the heavy chains are held together by the interchain disulfide linkage in the CH2 region, that is constant region of the heavy chain at 2 number position, which are near to the region known as, this region is known as the hinge region. This region is known as hinge region. This hinge region is more likely to undergo proteolytic cleavage. It will be cleaved by the proteolytic enzyme, and the portion of the antibody that has the capability to bind to the antigen consists of, that has the capability of binding to the antigen consists of variable region of the light chain and variable region of the heavy chain. So here also you'll find the variable region of the heavy chain and variable region of the light chain. This region after proteolytic cleavage containing the heavy chain which will interact with the antigen is called as fragment of antigen binding. Fragment of antigen binding. And the region which is not binding, the region that is below the hinge region after proteolytic cleavage that will not interact with the antigen is called as FC region, where this region is able to crystallize at very low temperature. This region is called as FC. The fragment of crystallizable region is represented as FC. Is the distal region of the antibody that is composed of two identical disulfide linked peptides containing the constant region of the heavy chains. Constant region of the heavy chains are held together by the non-covalent interactions that belongs to the heavy chain. That belongs to the heavy chain, like CH2, CH3 in few immunoglobulins, or it may be CH2, CH3 and CH4 in few immunoglobulin. FC region communicates some cell surface receptors called FC receptor, and that feature of FC region helps antibodies to stimulate immune system. So these are having the receptors on the adjacent cells which are involved in the immune reaction, and hence they transduce the signal they perceive by binding to the antigen at the antigen binding. So that's why this constant region is, or FC region is the effector region of the immunoglobulin, whereas the variable region is the antigen binding region, or it recognizes the antigen to carry out the, or to bring about the particular immune reaction. So this is regarding the structure of the antibody, and here you will find the looping of the each domain of around 110 amino acid residue, and these loops are formed by again disulfide linkages, and these disulfide linkages are within the light chain or within the heavy chain, and these disulfide linkages are called as intramolecular. They are called as intra-chain disulfide linkages. So here in the light chain you have the two loops, so two intra molecular or intra-chain disulfide linkages formed, and here in case of the heavy chain you will find one loop here and second loop, then third loop, and then fourth loop. So you'll find 4 intra-chain disulfide linkages, and the disulfide linkages varies with the number of the constant region in the heavy chain. So this is the FAB fragment which interacts with the antigen, and this is the crystallizable region which transduce the signal to the adjacent cells or to the other mast cells or the dendritic cells to carry out the further immune reaction. So this is regarding the antibody structure. Next, we will be using the term monoclonal antibody. Monoclonal antibody. So what are these monoclonal antibody? So till now we learned the structure of the antibody, which is Y-shaped structure having the region for binding the antigen and transducing the signal to the other adjacent cell or to the other lymphocyte to bring about the immunity. So now you should know the difference between the polyclonal antibody and the monoclonal antibody. When you have the antigen, when you inject this antigen into the host cell, when you inject this into the host cell, the surface of this antigen will be having these specific molecules. Surface of this antigen or the foreign substance will be having the specific molecules, and these will be recognized by the host cell, the lymphocyte, and they start producing different types of the antibodies, and each population or each antibody will be specific to this specific antigen or the antigenic determinant or the epitope that is present on the surface of the antigen. So if we just imagine this is the A type of the epitope, and this is B, and this is C, and this is D. Now if you inject this pathogen into the host, you will have the antibodies like antibody which will interact with the A epitope, you will have the antibody which will interact with the B epitope, you will have the antibody interacting with the C epitope, and you will have the antibody population interacting with the D epitope. And this host organism carries all these antibodies which will recognize this antigen at all the different specific sites, and these antibodies are called as the polyclonal antibodies. So here is an example where you have the different antigenic determinants, and these will be recognized by these antibodies at the different site on the surface of the antigen. So this combination of antibodies which recognizes the same antigen but at the different epitopic region or the antigenic determinant, these are called as polyclonal antibodies. And at the same time, you can also detect this pathogen by using only one antigen or the one antigenic determinant or only one epitopic region. Such type of the single population of the antibodies which bind to the antigen at the particular epitopic region, they are called as the monoclonal antibodies. They are called monoclonal antibodies. So the concept of monoclonal antibodies was given for the first time by George Kohler and Cesar Milstein in the year 1975. These monoclonal antibodies are the antibodies that are specific to one particular antigenic determinant, as they are made by identical immune cells. They are made by identical immune cells. That are several copies of a same parent cell. Example, the tumor cell of a specific region, say plasma cells, are monoclonal and thus have the ability to produce the antibodies of the same specificity. So the monoclonal antibodies will be having the same specificity in a population of cell. The basic technique involved in making of monoclonal antibodies relies on the fusion of the B lymphocytes from an immunized mouse with the myeloma cell, that is the tumor cell line, and that is called as hybridoma technology. So this technique is called as hybridoma technology, wherein the activated B cells are fused with the tumor cells. These tumor cells or the myeloma tumor cells have the capacity of producing only one specific type of the antibody. So the B lymphocytes, when they are fused with these myeloma cancerous cell lines, they produces the monoclonal antibody, and this fusion is called as hybridoma technology. And these monoclonal antibodies are advantageous and they are having the pharmaceutical importance. The monoclonal antibodies help in immunodiagnosis of various antigens or the antibody. The many tumor specific antibodies help in detection of the cancerous condition. Some of the monoclonal antibodies have the therapeutic uses. Example, CNF, that is a cytokine tumor necrosis factor. It's otherwise called as the TNF. The tumor necrosis factor is used to treat many inflammatory conditions. And similarly, the monoclonal antibodies help in identification of individual cell populations. Example, lymphocytes and leukocyte differentiation has become possible now because of the monoclonal antibodies. These monoclonal antibodies help in the purification of cells in order to generate the information about their features and their functions. So that is regarding the advantages of monoclonal antibodies over the polyclonal antibody. So in this session, we learned what are antibodies, what is serum, what is antibody, and what is serology, and we learned about the structure of the antibody. and we know the difference between the polyclonal antibody and the monoclonal antibody, and we glanced on the few functions performed by monoclonal antibody for the human betterment. I hope you understood this session. Thank you.